Hello, I am Ewusi Shiburah, a graduate student at the Institute of Biosciences, UNESP. And today I'm presenting on the knockout of the telomerase reverse transcriptase chain in Leishmania parasite and how it influences negatively the growth and cellular profile of the parasite. According to the World Health Organization, close to 2 million new cases of leishmaniasis is reported annually. Unfortunately, there are no effective remedies for this disease. This is why we chose to study the telomerase reverse transcriptase chain in order to provide new therapeutics against leishmaniasis. Our goal is to understand the role of this gene in the parasite. It is known that the telomerase reverse transcriptase gene helps in genomic stability and chromosomal integrity. So using the CRISPR-Cas9 approach, we do a lot of function studies and here we show some of the key methods employed in the study. We first generated Leishmania cell lines that were constitutively expressing Cas9 gene and T7 RNA polymerase gene. From there, we designed single-guide RNAs and donor templates to knock out the TERT gene and replace it with a neomycin donor template. We followed it up by doing a single-cell cloning, which was then taken through confirmatory studies and subsequent effect studies. Here, I show a schematic of the knockout procedure used in the study. I must mention here that all data presented herein were carried out with at least 3 biological replicates, and appropriate statistical treatment were meted out to each and every data presented here. So the first result we have here in figure 1 is the constitutive expression of Cas9 as detected by Western blot, and the JPDH as our loading control. Having observed the constitutive expression of Cas9, we went on to do a growth curve analysis of this parasite against the wild type cells, and we found no significant effect on the growth, just as was reported in 2017 by Beneke and colleagues. We went further to study the effect of the Cas9 gene on the Leishmania cellular profile. Once again, we found no effect on the cellular profile of the parasite, indicating that the Cas9 had no effect or whatsoever on the parasite. With this positive result, we moved on to carry out the main objective of the study, which was to knock out the telomerase reverse transcriptase gene by first amplifying the single-guide RNA in the donor template, which were used in the transfection procedure. In figure A is the donor template, and in figure B is the single-guide RNAs. After successful transfection, cells were selected by single-cell cloning via limited dilution on solid plates. We then took these cells through a confirmatory PCR diagnostic study where we found that the TERT had been knocked out of the parasite, and in its place was the neomycin insect using various primer combinations as shown here. We went on again to do a confirmatory study by primer walking along the TERT locus using Sanger sequencing, and we found that indeed the TERT had been knocked out of the genome of the parasite, and the locus had been replaced by a neomycin sequence as expected. With this positive result, we then went on to do a characterization of the TERT knockout cells against the wild type. Here, we first started by looking at the growth profile of the parasite, where we observed a striking difference between the growth of the wild type as against the TERT knockout. And this striking difference led us into looking at the apoptotic rate of the parasite. The apoptosis analysis showed an increase in the apoptotic rate of TERT knockout as against the wild type cells, similar to data presented on TERT knockout in cancer cell lines. Concurrently, we found a significant cell cycle arrest in the S phase of the TERT knockout parasite, and also an increase in DNA damage signaling. This corroborates data presented on TERT knockout in yeast and cancer cell lines. In conclusion, we say that it is possible that a TERT may play a significant role in the DNA damage response pathway and cell cycle progression beyond just telomere maintenance and genomic stability roles as ascribed to it. We hope to find more data on the effectivity of the parasite by looking at the TERT knockout against the wild type again. Once again, I'm Ewusi Shiburah, and I thank you for your attention.